there are two online meetings happening right now. This is obviously the right one. So let me go into the other one real quick and tell them to stop that. Okay, I'm back. All right. Well, it looks like a lot of you have been throwing up your candy phylogenies. Let me share my screen. And let's take a look at some of those. If I can find them. There they are. Okay. So major points for creativity here. We've got a little timeline thrown in, appearing to use real data to construct a phylogeny. But I don't like it, because it's not a phylogeny. That's okay. All right, this one actually looks like a phylogeny. Although it's interesting that the banana being the common ancestor to all of these took so long for banana to re-evolve. All right, here is also a not phylogeny. Another not phylogeny. What's the common theme here? I was hoping you guys would make this mistake, actually. You did not disappoint. It's a very common mistake when people are first learning about what actually is being displayed in a phylogeny.
Is it like the previous like ancestor sort of things? They're like putting them at the like in between the. Sorry, I don't know how to describe it very well, but
you're describing it pretty well. Like Hershey's dark chocolate evolving into milk chocolate. Is that what you're?
Yeah, that sort of thing.
Like the in-between stuff. So what this is showing is that there was some common ancestor of these two major groups. It looks kind of like maybe a chocolate group, except for the blow pop, and then like a fruit group over here. But then it's kind of just like picking up the dark chocolate Hershey's along the way. Okay. As opposed to, let's find yeah, like this. So what's the difference? What's the theoretical difference between those two drawings we just looked at, this one and the previous one?
It doesn't go from one state to the other linearly. It like every time it branches off and divides between two different things or two different directions.
Yeah, two different groups. Here, what this phylogeny is actually saying is that Hershey's is extinct. Does that make any sense?
So you wouldn't show any current living species on an actual phylogeny. It would always be common ancestors.
Yeah, so all these branch points, they're not living species. They're gone. They diversified into 2 new groups.
But for example, banana is living, right, in this case?
Yeah, banana's living. It can't be the common ancestor of all these things. So every semester that I do this experiment, I get a lot of phylogenies like this. Basically, and reading this as a biologist who looks at these a lot, I'm saying, oh, Laffy Taffies aren't around anymore. They diversified into this flavor and that flavor airhead. Okay. So what this is saying is that there's a common ancestor to all these groups that we see. The branches are groups. Okay. Usually on a phylogeny, they depict things that are still around, still extant. It's the opposite of extinct. Okay. But it's definitely a defined group that we know about, we can study in some way. It's usually got a name, something like that. Whereas these nodes, the branch points, what do they represent?
Common ancestors?
A common ancestor, and this is important, a theoretical or hypothesized common ancestor that we don't have an example of. If we did have an example of it, then it would get its own branch. I feel like maybe I'm not explaining this super well. So I'm going to go into our case study here again.
So is that
Yeah.
is that saying that like even if the like common ancestor was like extinct, but we know what it was, it would have its own branch?
Yeah, absolutely.
But would it still be shown as a common ancestor?
No, because it literally couldn't be, because it was its own thing. This might make more sense here in a second.
Okay.
Yeah, because we'll look at like fossils, and that's a good example of, I think, what you're talking about. All right, so we started with this kind of case study. People dying of MRSA. It's like making their feet rot off in a hospital, and researchers want to trace its nearest relative, MRSA, to figure out what to treat it with. So you can build a phylogeny, okay? And let's look at this again, just kind of in review. These branch points are common ancestors, hypothetical common ancestors. The tips of the branches are called taxa. Usually this kind of means like species, but it could also mean genus. Kind of depends on the resolution of what we're looking at. If you're an epidemiologist tracking COVID strains, for example, you're not even looking at species. You're looking deeper than that. You're looking at like strains of COVID. And so those branches are discrete taxonomic units of some sort. Okay. We got a couple different kinds of branches here. And I saw some of you do this when there's more than 2 coming from the same branch point. We call that a polytomy. What's that mean? Think in like biological terms. What would it mean for one ancestor to break into 3 evolutionary groups?
Basically, isn't it where like they don't actually know which came first, like which broke off first and then which kind of followed, and so it's kind of like a gray area?
It means a gray area. It means we have not resolved which one broke off first. Yeah. By definition, these do have to be bifurcating trees if they're to be quote unquote real. But oftentimes we don't have good information. We're missing some data. We know that these 3 are closely related to each other, but we don't know which 2 of them are more closely related to each other than the 3rd. Okay, so that's what's being shown here with taxon A, B, and C. B is more closely related to C than it is to A. C is more closely related to B than it is to A. So we can look at this group and say that they are sister to each other. with that in mind, what is sister to taxon A?
Both B and C?
B and C equally. All right. We do this by basically just kind of like backtracking down. All right. We find, okay, A, how long does it take to get to a common branch point? Here. Okay. There it is. Now, the same distance along a common branch point the other direction, B and C are equally distant from A if you were to track this back and kind of like measure it with a ruler, so to speak. We can actually do measurements by looking at the number of bases that change in a genome, that kind of thing. But for us, for this intro class, think of it as like measuring with a little piece of string back to the branch point from the taxon you're interested in. Take the other direction from it and then see which ones have the same distance. All right, does that clear up that? Taxon A's sister is B and C. Okay, what's the sister to taxon D?
E and F.
You would think E and F, but we actually don't know, and that's what this is. When we come back here, we don't know which paths to take. Here we find the branch point, and we have a certain path that we can take, and we take all the stuff down that path. But here, since we aren't sure, it's unresolved. And so sister to taxon D is E or F, or E and F. We just don't know. Because it could be that D and E are actually sister to each other, and the F is kind of the oddball out, or it could be the other way around. Okay. How many descendants came from this branch point node common ancestor?
Is it 4?
3. So everything downstream from this point, A, B, and C. How many descendants came from this ancestor?
6.
6. Great. All right, so we're starting to get the feel for this.
So in a phylogenetic tree, then, will there only be like labels at the very ends of all the branches? There won't ever be like a label in the intermediary like points?
Yeah. There won't be a label right here in the middle saying, you know, dark chocolate Hershey's. Okay. Because we know about dark chocolate Hershey's. It's not a hypothetical organism or candy. It gets its own branch.
So if we have a pretty good idea of what the common ancestor is, do we ever write that or no?
Yeah, we sure do, and that's subject to a lot of, you know, evolutionary research is hypothesizing about what this branch point here would have been like.
But we wouldn't write that in a phylogeny, or do we just say that's extinct and clarify that?
You could, and I've seen this in papers.
Riley?
They can do like a little note, kind of like this says branch point node and points to it. You could say like had fins. Point here, yeah.
Okay.
Yeah. All right, so I asked you to make a phylogeny. I'm going to show you the one I came up with. It's not right, because there is no evolutionary relationship between these candies. But this is the one I came up with. I have banana here as the outgroup. What does outgroup mean? What's the point of it?
It means that it doesn't quite fit in with the rest.
It's there to show you that the rest of your stuff is monophyletic. So a phylogeny of candy, a banana is not a candy, arguably, I guess. But all of these are. And so the banana is kind of the weird one out, and it's closely related to the stuff you're actually interested in, but it's not part of that group. And so we call that an outgroup. So I had the common ancestor of bananas and candy. I could make a note here and say like, oh, this common ancestor probably had you know, sucrose or fructose as part of it. I don't know what else I could say that's similar between those, but then we branch out into kind of a fruit group and a chocolate group. Okay, so we could draw a circle around just the fruit and say this is a monophyletic group called the fruity candy. We could do the same for the chocolate. Say this is monophyletic group of the chocolatey candy. Let's take a look at a few phylogenies that aren't neat and tidy like this with everything on one line.
Monophyletic meaning what?
Meaning everything downstream from a common ancestor. So everything from this branch point, if I looked at that whole group and circled it, that would be monophyletic, meaning one phylogeny. They all go back to a common thing.
So is that a clade, or what's a clade?
A clade is any group that you want to draw. We'll get to that in 2 slides. But first, I want you to tell me which one of these phylogenies is different from the other 3.
Would it be the first one in the upper left-hand corner? Because it's just neatly, and the rest of them kind of have all the triangles and weird stuff going on.
Looks like the bottom left one is.
Yeah, the bottom left.
Bottom left.
Why bottom left?
Because they all come from points instead of having.
The suckers branch off later than in the other ones.
Ah, okay. So yeah, you got to get past kind of the surface level of how the phylogeny is drawn and look at that it's showing hypothetical relationships between these species or groups. And here, the suckers are kind of out on their own. They branch off from the rest of these early on. Same in this one. They branch off early on. The airheads are sister to each other. The Laffy Taffy are sister to each other. Airheads, sister. Laffy Taffy, sister. Laffy Taffy are sister and airheads are sister, but the branching order is different. So if I was to redraw this phylogeny in the lower left, like the one in the upper left, the Laffy Taffy and lollipops would basically switch places. Does that make sense? It gets confusing when you look at phylogeny. Sometimes phylogenies are drawn like this. If you get a lot of taxa, they put them in kind of a circle. Think about the relationships that are being depicted in terms of what is sister to what. So top left, the sister group to the lollipops is what?
All the other ones, really.
All the other ones. Right? Go back the lollipop group, go back here, then everything downstream from that is sister. In this one, what's sister to the lollipops?
Airheads.
The airheads. And so they're depicting different relationships. Usually we're going to try to look at phylogenies that look like this. Nice and tidy, kind of easy to read, but you need to be ready for it. Even the textbook has some circular phylogenies, and we'll have to see some coming up. Now, we had a question about what is a clade. So on this, I could say that banana is a clade, blow pop is a clade, Tootsie Pop is a clade, or lollipops is a clade. So it's any group that you, the human, want to try to designate. So I'll show you some clades, and this is how things get named. Okay, you see ACE, that means family. So Fruitaceae, this is just a made-up word I made for the family of fruit. And the Cocoaceae, this is the family of chocolatey stuff. I can refer to the Fruitaceae Clade, and if you had this phylogeny, you should know that that's everything downstream from Fruitaceae. So that'd be all these candies here. I could have the Guminosus Clade, and that would refer to airheads and Laffy Taffy. So the term clade just refers to some branch point and everything downstream of it. Kind of depends on what level you want to talk about. In this group, what is the candy clade?
That would be just like right above the Fruitaceae and the Cocoaceae.
Yeah, right here. So this could be the phylum Candy versus the phylum Fruit, for example. Now, some clades are monophyletic, some are paraphyletic, some are polyphyletic. This is a monophyletic clade, Guminosus, and that's everything downstream from the branch point, including all these hypothetical ancestral species. Okay. So is Linguadenta monophyletic?
Yes.
Yes. Okay. Is Butterfinger monophyletic?
That would be poly, right?
Let's take a look.
Butterfinger by itself is monophyletic.
By itself, it is monophyletic. Yep. Now this is paraphyletic grouping. Paraphyletic meaning that I've got the common ancestor of Fruitaceae, but not all of its descendants. Okay, so for whatever reason, me, the human, when I looked at this group, I left out the banana Laffy Taffy. Now, do you know why I left out banana Laffy Taffy? Because it doesn't qualify as real candy. It's a foul spawn of Satan. Okay, so I, the human, judging candy, let's say I look at these and without knowing their relationships, I say, okay, well, here's the legitimate fruity candy. It's the Tootsie Pop and Blow Pop and the Airheads and the red Laffy Taffy. Great, and I'm leaving this one out. Well, I've made an evolutionary mistake by doing that. It means that I didn't realize that the banana Laffy Taffy is actually part of the Fruitaceae Clade, and so it's a paraphyletic group. Now, polyphyletic is pretty much the same thing. I'm not too concerned about the difference between para and polyphyletic. Polyphyletic is just when you don't count the ancestral states as well. And so we're just looking at these groups here. Okay, so it's the same kind of mistake, evolutionarily speaking, as paraphyletic. I'm much more interested in the distinction between monophyletic and not monophyletic.
So if you were to start at the common ancestor for fruit and then all candy, that would technically be mono? Or would that be... Is that would
like
include...
if I was here at this branch point?
No, one more up.
Oh, right here.
Yeah, would that technically be mono or not?
It would be monophyletic if you counted everything down here at the end.
Okay.
And so it's like we're trying to group together all these branch points, the tips of the branches, and call them some name, whether it's a genus, a species, family, class, order, phylum, kingdom. Those are all groups that it's just humans kind of grouping things at some arbitrary level, essentially. Now, the mistake you don't want to make is accidentally leaving out some of these tips that are part of that group. And so if you went to this group at the very top, this branch point for all the candy and the banana, and you said, this is candy, what kind of mistake would that be? It'd be
Paraphyletic.
paraphyletic kind of thing, right? We're leaving out the bananas accidentally. We're really, this branch point down here would be the monophyletic branch point for candy, and as long as we included all these candies. Now this has happened over and over. It's still happening when people make phylogenetic trees. Okay, we think of a group of organisms as being something, right? Like pine trees. Well, these all look like pine trees, so we're going to call them pine. And the Latin name for that is pinus. Okay, well, it turns out when we do DNA sequencing that some of these pinus are actually more closely related to other trees than they are to the rest of pines. And so we've accidentally put them in the wrong group, and we've not got a monophyletic group there.
So paraphyletic and polyphyletic is actually just it's just a designation of an error.
Yeah. It just means the mistake you're making is pretty much the same with either one of those. It means that you're accidentally leaving out at least one descendant that should be in that group. Okay. Well, how do we make a phylogeny? I mean, here, this was easy. We just kind of like got creative and grouped things according to their taste or their shape or their ingredients, that kind of stuff. But in real life, how do you do it?
Phylogeny is based off kind of physical characteristics, isn't it?
Can be. So this one here is based off physical characteristics. And so if you got lancelets, lampreys, tuna, salamander, turtle, and leopard, you can measure a bunch of physical characteristics, some traits, like hair, amniotic, like a shelled egg, 4 walking legs, jogs, and a vertebra. Okay. So we look at those, and we make basically a little matrix comparing how many of these traits are similar between all these groups. Okay, so looking at this little matrix here, this character table, and your textbook will walk you through this. I'd recommend you kind of go through that part of the chapter and see if you can do this. Leopard shares some things in common with all of these, right? It's got a backbone, okay. It's got jaws, all right. It's got 4 legs, yep. Amniotic egg, yep. But then it has hair and the rest don't. Okay, so what's more closely related to that? Well, this one shares 4 of those same characteristics, the turtle. The only difference is that it doesn't have hair. Okay, so those share 4 characteristics with each other. That's pretty closely related. All right, then leopard and salamander. Okay, it doesn't have the amniotic egg, so it's only got 3 characteristics in common with the turtle and the leopard. So sister to the salamander, it's turtle and leopard because they both share those same 3 characteristics with the salamander. But the turtle and the leopard share 4 characteristics with each other, and so they're more closely related. It's a bit confusing. So if you have any questions, please let me know.
So how would we know that like the lancelet has this common ancestor since it doesn't share any of those traits?
It's the outgroup. It's the weird one that doesn't share any of those traits. And so we just put it there to kind of look at the relationship of these to each other. It gives us a monophyletic group of lamprey, tuna, salamander, turtle, and leopard. By its nature, the outgroup is something that we're not interested in or maybe don't know enough about.
Okay.
So is there always going to be an outgroup then, just so we have that kind of like similarities between the rest?
Most trees, you don't have to have an outgroup, but if you want to have it what they call rooted, you'll learn about this in like your evolution and phylogenetics class. if you want your tree to be rooted, you do have to have an outgroup, and that gives you some mathematical properties you can play with doing analyses.
Okay,
So
thank
it's
you.
most common to see an outgroup.
Is there a danger in doing it this way? Since, I mean, you could arbitrarily pick, 10 characteristics that they all share.
I'm giving you a non-ironic slow clap. Love it. Yes, it's very dangerous. Because why did we pick those 5 traits? Hair, amniotic egg, 4 walking legs, jaws, and vertebral column.
Well, it makes it convenient to make the chart.
Yeah.
So like let's say humans were on that list. Would they go next to Lancelet, or with the outgroup, or where would they go?
They'd go next. They'd be up there with leopard because they also have hair. And so leopard and human would be the sister to each other.
Okay, yeah, I see that.
Yeah.
Would they offshoot because of the 4 walking legs and we're bipedal, or would we still be next to leopard?
We're actually not bipedal, technically, according to tetrapod rules, and that's what they're talking about here. We've got 4 limbs, 5 digits on each limb, so do turtles and leopards.
Gotcha.
Yeah. Okay, and then if we threw chimpanzees on here, we'd have to add more traits to start telling which were more or less closely related to leopards or humans, right? Because chimpanzees, leopards, and humans all share these 5 characteristics, so there's not enough data to resolve those 3, and so we would have a polytomy, right? How we get out of that is to look at more traits.
So for example, opposable thumbs.
Right, opposable thumb. You could add that in, and then boom, you've resolved that trait. But, like somebody just brought up, this is kind of dangerous because we're just picking characters, and it's limited by our imagination, our inherent biases, all that kind of good stuff. Right, so these are morphological traits, but we can also use molecular traits in the exact same way. Okay. So when we're looking at phylogenies, I mean, what's the point of looking at a phylogeny other than this academic exercise of seeing what's sister to a leopard?
We could possibly understand how certain things evolved, maybe, based on similar traits like amniotic egg or hair.
But who cares?
The MRSA patients.
How does
the MRSA patients? Sure.
How life evolves. I
mean, that's just knowledge for knowledge's sake, unless you're applying it somehow, right? So this phylogeny, sure, we could look at this and like learn it as introductory biology students, but it still doesn't tell you anything useful. What it does do, though, is it makes predictions. Every single phylogeny you've ever seen is a hypothesis. Keep that in mind. And hypotheses, their job is to generate predictions. Okay. Predictions that can be tested. All right, so here's another phylogeny. We've got birds, saurischian dinosaurs, ornithischian dinosaurs, crocodilians, lizards, and snakes. Okay, according to this phylogeny, some of you probably already have misconceptions about the relationship between crocodiles and lizards. What does this tell you about that relationship?
It tells us that lizards and snakes are less closely related to crocodiles than birds are.
Yeah, birds are more closely related to crocodiles than crocodiles are to lizards, which is galaxy brain, right? That's weird. And so what it also means is that when we say birds, that's not monophyletic. If you say birds, to be monophyletic, you also are including crocodiles, which everyone knows is not a bird. It's a crocodile. But this is the weird kind of stuff that we get. This is generating a hypothesis. It may be hard for you to believe, but it's making predictions. So one of those predictions, crocodiles being more similar to birds than they are to lizards and snakes. Okay, well, let's look for that. So we can go to these extinct things, Saurischian dinosaurs, and say, okay, if that's sister to birds, then they're going to have a lot of traits in common that aren't shared by crocodiles or lizards and snakes. Okay. And so we make a prediction, and then we go out in the real world and we test that prediction. We look for evidence to disprove it or help us confirm it. Okay, well, these dinosaurs do have a lot in common with birds. Their bone structure is the same. We found that they laid eggs and nested on them like modern birds do. Okay. So we tested that prediction, couldn't disprove the hypothesis. All right, so keep that in mind. These are hypotheses, they generate predictions. This is a hypothesis, it generates some predictions. We may not trust this hypothesis because it was built on this little character table. So if we want to trust it more, we go to molecular data. Now, keep this character table in your head and look at this. This is a DNA alignment. So for some gene, we sequence it and we align it with a bunch of different species of interest or strains of interest or what have you. And then we look down each column and we look for things in common, exactly like this process. So kind of flip this around so that the characters are on the top and the taxa are on the left-hand side, and that's what we've got here. Each base pair, each position for the nucleotide is a character. And so instead of saying, do you have hair or not, we say, okay, in position 1 for this gene, do you have an A? Yes or no? For position 2, do you have a G? Yes or no? Does that make sense? Or not? We're counting up the number of shared nucleotides at each position for 2 taxa, and we're saying, okay, what's your relationship? How distant are you in terms of this? And we give it like a little mathematical number of, here's the changes between this group and that group. And then we do this group and the third group, and then this group and the fourth, and the fifth, and so on, where we've made comparisons between every possible pair of the species we're interested in across all of those DNA positions.
Now,
Oh, you go ahead.
Does their DNA being the same or very similar necessarily mean that they're closely related, or is there a possibility that 2 different disparate branches of evolution happened to create the same DNA?
Yes. To the second part. It is possible that convergent evolution pushed 2 groups to have the same letters in the same order right there.
And isn't that why it's important to just look, not just look at one or the other, but you've got to look at like the whole picture. You got to look at the morphology, the DNA, and everything
else.
So
we look at the fossil record, we look at character traits to kind of give us a sanity check on our DNA-based phylogenies. Also, the more DNA you use, you're exponentially decreasing the likelihood that that happened accidentally. Okay. So we've got 4 possible letters at each one of these positions. What are the odds that 2 groups have the exact same bases, nucleotides at those positions on accident? Well, if it's one position you're looking at, the odds are 1 in 4. If it's 2, what are they?
Want to be 1 in 16?
1 in 16. If it's 3, boom, exponential. And so if you get up to like 1,000, 10,000 characters that you're looking at, we can be really, really confident that it wasn't because of accidental convergent evolution. Okay, so this little case study about the MRSA patients wasn't just made up, it was a real thing. And they did a DNA alignment, they built a phylogeny, and here's what they found. Okay, these are the Thailand cases out here that they were worried about. What do you notice?
They're monophyletic. They share a common ancestor.
The Thailand cases don't. They're color-coded.
We've got 2 cases from outside of Thailand, one from the UK and one from Denmark.
One from the UK and one from Denmark that are actually more closely related to the Thailand strains than they are to other European strains. Okay, so here's Australia, here's the USA in black, here's Greece, Turkey, Uruguay, Brazil. So we've got like a South American strain, we've got Southern European strain, and then we've got Chinese here and Thailand. And so researchers were initially thinking it's going to be most closely related to the Chinese strains because Thailand, China, very close. But turns out it wasn't monophyletic. They had a paraphyletic grouping here, and Denmark and the UK were in there. All right, so the hospital got in touch with the hospitals in Denmark and the UK, and it turns out the strain actually did come from the UK. And so they were able to successfully treat this outbreak with the right medicines just by doing a phylogeny based on a DNA alignment from these strains of MRSA. Okay, so that's
You're muted.
Hey, Jeff, you're muted.
Can you hear me now?
Yeah, we can hear you now.
Yep.
Okay.
Hear ya.
That was weird. Can you see my screen still?
Yeah.
Yeah.
Okay. So this is a hypothesis that we're looking at. It made a prediction about the relationships of these strains, and one of those predictions was that the Denmark strain from 2001 or the UK strain from 2003 is probably the most likely source of this new MRSA infection. So it made a prediction, we went out into the real world, tested it, and yes, the drugs that were able to treat the UK infection were able to treat this Thailand outbreak.
Would all of that be computer generated? So would they just take?
Oh yeah.
Would they just take the DNA strands and then put it into computer and it would put out the phylogenies?
Absolutely, and we're talking about millions and millions of comparisons. Like, you can't look at this as a human and have time before you died to like count up all the differences between these different strains across this long sequence. It just wouldn't happen. Yeah, so computers are absolutely necessary for this kind of work.
How often are these phylogenies accurate and correct, since all of them are technically hypotheses?
Well, none of them are accurate and correct.
So why do we rely so heavily on them?
Well, here's the thing. The law of gravity is also not accurate and correct. But it's good enough. It approximates reality so closely, and it passes every practical test that we throw at it that we just go ahead and use it. That's what we're doing here. And this is a hypothesis. It can be changed. If we get more data, it's probably going to change. But it worked well enough to save the lives of these people in Thailand.
So basically, we just use these speculations and hypotheses to get functional data that we can use.
To get functional predictions.
Yeah.
The data is what made the hypothesis. The predictions, what a hypothesis is good for. Something we can test. Okay, so this survived that test. And that's a big test. The lives in the balance kind of test. And it won. So we know that this hypothesis is at least good enough to answer our question. But it's not reality. Now, what about this? Does this look familiar to you now? What are we looking at here?
It's a phylogeny.
Yeah, it is. Now, something special about this phylogeny is that this one, the first one that I know of, that's not a hypothesis. Because we saw it. We saw it happen in real time. And so this is a real phylogeny. That's very rare. That was the point of this study. They made a real phylogeny, and that's why this study is so awesome, is that we know the actual phylogeny of this event, okay, of these strains acquiring antibiotic resistance. And so when we make predictions from this phylogeny, we have the reality to check those against 100%. Every prediction we make can be checked against this real phylogeny. Okay. This is awesome. All right. This is what biology is all about. This kind of experiment just blows my mind. They documented this real-time phylogeny with full genomes all the way as it's acquiring antibiotic resistance. Now I got a question for you. What if we rewound the clock here and started this experiment over? Exact same conditions. What would we get?
Probably something different.
In what way?
Well, I mean, if this happens because of trillions of dice being thrown every moment, it would just be different results, wouldn't it?
I could almost
the same thing would eventually happen in the terms of it, like eventually overcoming the antibiotic, but it'd go about it in a different way.
Yes, it still would overcome the antibiotic, but it would do it in a different way. Absolutely, because the mutations are random. It might not have been, this particular strain that made it in first. Maybe up here is where the breakthrough would have been. Okay. And so that's why they did this simultaneously on two different sides moving in. We get these different phylogenies basically moving towards the same kind of end goal that was set up artificially by the people who made this experiment, but we get to see how likely it would be that the same genetic pathways would be involved in overcoming resistance or overcoming this antibiotic. And they were different. These groups over here, this group up at the top didn't even quite make it. These groups here and these groups here, the orange and the red, they acquired their antibiotic resistance through different molecular pathways. And there's no guarantee that that would be the same if we rolled the dice again because of the randomness. So there was a famous biologist, Stephen J. Gould. He's a paleontologist, wrote a bunch of really hard to read books. He did a lot of work in this area in Canada called the Burgess Shale. And this is really cool formation because it's just exposed and it documents animal evolution through the Cambrian and on for millions and millions and millions of years. Okay. And he wrote this book called The Burgess Shale that was basically asking this question: what if we rewound the clock, went back to the Cambrian, what are the chances that we would get the same stuff now? Well, pretty low. quite low. But what are the chances that we would get eyeballs? Pretty high. Those have evolved independently 41 times. So it's kind of like saying, okay, yeah, we're going to get some features like resistance to the antibiotic. We're going to get some features like locomotion, being able to move around, that happened repeatedly during the Cambrian. We get features like the ability to use photons to understand your environment, so eyeballs, kind of, right? We get the evolution of flight repeatedly, because that's a whole new thing that you can move around in, but there's absolutely no guarantee that this would happen the same way.
So would an implication of that be like if like a whole biosphere evolved on a different planet that was similar to Earth's, that they may look different, but they would probably still have fins and wings?
Yeah, they'd probably still have ways, if it was the same Earth, right? And it had water, salt water, an atmosphere, all that kind of stuff. Yeah, we'd have wings, we'd have some way of getting around. If sunlight was a thing that was important, which it's gonna be, we'd probably have eyes or some way of responding to light and dark. But it wouldn't necessarily be the dominant creatures having 6 limbs, like insects, right? Maybe they'd have 20. We've got this little weird fossil here called Hallucigenia. It went extinct. Nothing after it had its same body plan. It just didn't make it. But what if it didn't go extinct, right? What if it became the foundation for all the creatures that we see around us now? You and I would have more limbs, probably.
So you said this was a book that he wrote, the Burgess Shale.
Stephen J. Gould wrote this book. It's not on our list because, man, he's a good writer in the sense that he thinks everyone should be able to speak French, and so he puts long, like 3-paragraph sections in French. It's just really pompous, but it's hard to read. You kind of have to have like a Google Translate app next to you. I didn't make it through the book. Gotcha. I read his papers instead. His scientific papers are easier to read than his books, which is saying something terrible about his books.
Okay.
Yeah. But it's still, for some reason, because of the ideas it presents, it is a classic in biology, the Burgess Shale. All right. We're out of time. You got a due date tonight for at least one assignment, and your quiz number 1. I've put a link on our main chat in the general somewhere here. I don't know where it went. Oh, right here. So there's another chance to meet one of our bioinformatics faculty candidates and hear their research and teaching presentation. Here's the link for it. It's 1 to 150 today, 10 points. If you write me pros and cons paragraph of what you liked, what was understandable, what wasn't, all that good stuff. Okay. I'll stick around.
10 points for everyone that you watch, or a total of 10 points?
No, there's 4 of them. If you go to all 4, that's 40 bonus points. This is unusual. I usually don't give out that many bonus points right up front.
And that doesn't fall under the your choice points.
No, this is bonus on top of everything. Okay. It's because I really do want to hear your impressions. We're hiring one of these 4 people. Okay. You're the one that's going to have to take classes with them starting in the fall. And so I want to know what you think.
What are the other dates that they'll be showing up?
Next Wednesday and Friday also.
Okay.
And those will both be at noon. And I'll put links to those.
Is there a way we can watch it if like we're busy during that time, like watch it after, like a recording or anything?
I don't know that it's recorded and distributed. I'll look into it. It seems unlikely.
I would like to do it, but I have a class, so
Okay.
So if it is recorded, I would like that information as well.
Okay. And keep in mind that there's next Wednesday and Friday will be at noon, and I don't know many classes that are scheduled at noon, so You guys can definitely make those two. If I can't find a link to the recording. All right. Well, I will see you next Wednesday. Have a
Thank
good 3-day weekend.
Thank you.
Thank you, Professor.
Thank you.
Hey Jeff, I just had a quick question.
Thank you.
Yeah.
Thank you.
So the pretest that's due today, I remember you mentioning that this one, as long as we take it, we get full credit. Is that just an illusion of mine?
No, that's right.
Okay, great. Awesome. Thank you.
Mm-hmm.
Have a nice weekend.
See ya.